I won't be able to, so it doesn't make all that noise. I think we should wait another few minutes. I think it was. Yeah. Of course. All right. Hi, everyone. If you are here for the shared resources, lunch and learn, town hall seminar, you're in the right place. We did have the grand rounds down the hall, so if that was what you were planning to attend, you can get up and leave. To start, we just have a couple announcements. So next month's lunch and learn seminar is also in a different room. When I went to book it, K307 was reserved already, so we'll be in Lower Adolph. I'll have it on the new flyer and updated on the event information, so you don't really have to worry. I just kind of wanted to call it out since we've been pretty consistently in K307. And then we have human biophys to kind of round out the rest of the shared resources. And that seminar is in October. So if you were submitting the follow-up surveys following the lunch and learn seminars, you were entered into a like a raffle drawing. So Christina Davidson had filled out 5 follow-up surveys. She's been pretty consistently attending the seminars, and after assigning each name of the entry to a random number and doing the random generator, Christina's name was drawn as the winner. So she'll win the $50 prize. And we're still going to continue the follow-up seminar survey so that we can improve the seminars as we continue to offer them. I think that was my last slide. Oh, and then upcoming later this month, early next month, we will be opening our shared resources calendar submissions. So if you are interested in submitting a pretty image, hopefully it's cancer relevant. We are going to do another 2025 wall calendar, and there will be prizes for selected entries. And we'll announce the winners at the Wilmot Symposium. So it's pretty cool. Good chance to show off some nice images of your work. And then our shared resource. Oh, I do have another announcement. Oh, sorry. In October, there is the CCAC retreat, which is the what does CCAC stand for? Community Cancer Action Council. And it's a good opportunity for you guys to present your research projects and talk with community members. So it's geared towards a lay audience. So it's a good way to practice communicating your science at a lower level and getting that high, big picture view communicated across to people who might not generally be aware of the research going on here at Wilmot. The abstracts are due Friday, September 6th. And then you'll be informed the following week whether your abstract was submitted. And the CCAC retreat is in Flom Auditorium this year. So it should be a good opportunity for more people to submit abstracts and present at the poster symposium. All right, and with that, we're going to launch our town hall seminar. So it's a good opportunity for a Q&A session with our director, John Ashton, and I'll let him present.
Thank you, Tracy.
There's no remote.
So just a keyboard. Okay. Okay. All right. Well, hopefully everyone was able to get lunch. If you don't, feel free to come down and get whatever you want during. This is very loud. So thank you all for attending the seminars. I think it's a real nice opportunity for us to kind of educate you and give you information on the shared resources. Today, it's going to be a little bit shorter. I just want to update you on some shared resources you might not know are available. And talk about why that is, and then have an open discussion. If there's anything that you think that you've either heard or you're interested in that we don't have, we can have those discussions too. We're always interested in feedback on what other resources might be helpful for your research. So briefly, most of you probably know who I am and you know who Tracy is from coming to these lunch and learns, but just to give you some context, both Tracy and I oversee shared resources at Wilmot. I'm not going to go through the details other than it's important that you have a face to a name, and anything that's shared resource related, even if it's not Wilmot centric, we can usually either help or point you to the right direction. We have a lot of expertise across the spectrum, and Tracy's been really great in managing and trying to sync up with researchers and shared resources need. So a lot of these lunch and learns have been focused on the core Wilmot shared resources. You've heard, if you've come to these, you've heard several of these already. There's an important distinction between the shared resources that are on the screen and the ones we're going to talk about today. Particularly, the shared resources that you've learned about and the ones I just showed are really designed to support the NCI Comprehensive Cancer Status designation. And so I thought it was important. Most of you probably have heard that we're, as an institution, we've submitted our application for this, and we're going to find out shortly how productive that was. So it's important to delineate all the shared resources you heard about until today are those core shared resources. Every Wilmot member has access to them. They have organizational structure. We track usage, we track acknowledgements, we give subsidies and discounts to those things, all supporting this goal to become a comprehensive cancer center. So what are the other things that we have? And there are reasons why these resources we're talking about today are not part of a bigger shared resource. We won't go into that, but if you're curious, I can chat with you afterwards. So again, basically on the left, you have a pointer. On the left are the ones we've always talked about. These are the CCSG-centric shared resources. Everything on the right are things that Wilmot has historically supported in one way or another that are available to the community. Oh, that thing is in the way. Let me just move this quick. Sorry. We'll talk about the Wilmot Animal Resource briefly, histology, which is something that we're going to talk about our collaboration with the Center for Musculoskeletal in supporting those needs, and compound screening. These are the three main other shared resources that are here, that have been here for a long time, and there is some utility to them from investigators. Our animal resource has been around for probably about 15 years, if not longer. The contact for that's currently leading that, her name is Liz Lemire. She's great. She's a very good animal resource director. She understands a lot about breeding and colony management and experimental services. Some of the things that she's currently supporting investigators are with colony management. So whether this is you have a strain of animals that you already have in your lab and you need help maintaining them, if you need help breeding them, if you need help with assessing how to cross certain lines to achieve a goal, that all falls under colony management. Breeding in-house, it's a separate entity. We breed some standard mice in-house to make them cheaper to investigators than Jackson Labs. A good example are Pep Boys or NSG mice, immunodeficient mice. They're very expensive if you order them from Jackson. We brought those colonies in-house to basically offer more cost-effective animal breeding prices. Some of them are listed here, C57, NSG, Pep Boys, and there are other ones. If there are demands for different types of in-house strains, those are things that you can discuss with Liz. She's very open to bringing in new lines if there's enough sufficient demand that it will help the community. And just for perspective, an NSG, a single mouse from Jackson, is around $200, $300. Liz basically charges about $50. So you can see the internalized cost where it's a much cheaper cost for those types of mice. Additionally, there's a lot of experimental services that she offers, from tissue procurement. If you need help, you have an experiment and potentially you just need help with taking the animals down, isolating tissue, she can help with that. Genotyping, if you have a colony and you have crosses and you need help with tail snips or certain ear punch related work to then do genotyping, she'll help with the processing and the genotyping, assuming that the assay is available. We also, through other shared resources, can help design assays if you need to help with genotyping. She also helps with cell transplants and dosing. So this is where there are certain investigators that will ask her to take certain cells and help generate a syngeneic animal model, for example. She can do the transplants, she can monitor the mice, she can help with the tissue procurement, she can help with the genotyping or the actual assay of the engraftment capability with maybe she'll bleed some of the mice and do some of the blood analyses. So really the point I'm trying to make here is that there are a lot of opportunities to interact with Liz in the animal resource. And there's a lot of things that she does Again, she's by herself. So, part of this is that we build based on demand. So the more demand that we have from the community, the more we'll be able to build out. In addition to that, she oversees the mouse blood cell analyzer. So it's a new HESCA. It's on the third floor of the Wilmot building. The majority of the use right now is the Wilmot third floor researcher community. But this is available if you need to do peripheral blood cell counts or other types of analysis on mouse blood. And also working with Brian Marples in the radiation imaging shared resource. She also oversees the mouse irradiation system that's in the KMRB. So if you need to do any transplants that require total body irradiation or sublethal irradiation, this is where you get access. Now, you've heard previously discussions around whether or not the gamma cell irradiator will go away because it's a cesium source. Our expectation is there always will be an irradiating system, whether it's X-ray or other, that will be in this facility that will allow you in the KMRB, which is a micro isolator facility, to be able to bring your mice and do those type of work. It's kind of small on the screen, but these are data from our recent Wilmot member survey asking about demand and need. And the takeaway from here is that all of these options really do emulate everything that Liz in this animal resource supports. So the community needs help with all of these things. We have the capability of helping all of those types of demands. And if you're interested in learning more, you can contact myself, Tracy, or I put Liz's contact up here. She's very nice, she's very helpful, and she can at least give you more information. Yeah, Brian.
What do you call animal protocol? Do you add into the protocol, or is there a standard way of accessing these services by your protocol?
So good question. So both, really. If you have a specific protocol that's unique to your lab, you put Liz on it, and then she's able to do that work with you under that UCAR. There are some protocols, mouse breeding, for example, that we have a universal UCAR protocol, and then we just transfer mice between protocols. There are some experimental protocols that we do a lot of for individuals, and so we incorporate that under ours as well. So we create a separate UCAR that's linked to the investigator. Yeah. Yep. Any other questions? Histology is one of the things that we've heard a lot that investigators need help with, right? It's basically being able to figure out how to section tissues, how to process tissues, and potentially how to get some advice on doing some immunostainings. Historically, Wilmot had a small histology core that was funded by Wilmot, and it was aligned with the Department of Surgery. Subsequently, through some retirements and other things, we decided to align with the Center for Musculoskeletal Research, their histology core called HBMI. And we did this for multiple reasons. One, they have a lot more experience in diverse sets of tissues and processing than our historical Wilmot histology core. Two, they have a lot more people. And three, it was a synergy where we were able to basically move some of the equipment that we had from Wilmot into that facility and allow them to give some cheaper prices to Wilmot investigators for histology. I'm not going to talk too much about what they can do because I feel like they're better at talking about that than I am. But I have put their mission up here and really some of the services they include. So they'll help with sectioning. They have cryostats. They have vibratomes, microtomes. They have embedding stations for paraffin or fresh frozen OCT embedding. They have a lot of experience and a lot of utility. And through our partnership, we're helping to provide some operational support so they can bring on more staff to help more folks. The one thing they don't currently do as steady state right now are actually the immunoassays themselves. So you cannot give them a section or ask them to make a section and do IHC or IFC. They do have experience consulting with you, so they can do the upfront processing and give you some guidance on how to do that, but they don't currently offer that as a service, and that's something that we're working towards. But that's pretty much what I've said here on the slide. These are the things that they have available to you. So if you have histology needs, I would strongly suggest contacting Jeff Fox in the HBMI core. Tell him you're a Wilmot member. He helps prioritize some of the work that you do as well as part of our agreement and relationship. Just staying in line. This is something that has been highly demanded over the years when we ask our membership at Wilmot, what do you really need? And histology has been a big ask. And again, a lot of the things that folks have asked to have in the histology facility are currently available. aside from the immunoassay support. And lastly, for those of you that might be interested in high content screening, compound screening, we do have the capability of doing that as well. This is really more important for those interested in drug discovery or candidate discovery, or if you have a small molecule library of a couple thousand molecules that you want to test in a cell model, the efficacy of that. And so I'm just listing here. So I'm the contact. So it's a Wilmot shared resource, but it's embedded in the Genomics Center, largely because of just the experience I have doing this type of stuff. So if you have any questions, feel free to contact me. Isaac Harris, who's also a Wilmot member, who some of you may have known, also helps with this. He runs it a lot. He does a lot of specialized libraries, and he's really helped us basically update the platform and make more utility out of that platform. But still contact me first so we don't overburden Isaac. These are some of the collections we currently have. We have several small compound collections. Again, part of the context here is if you wanted to screen FDA-approved compounds in a cell line that you have a specific reporter in, you have the ability through the Spectrum collection to do that. If you wanted to just look at different structure function by screening a bunch of different molecules that have different structures, we have the Cambridge Diverse Set, which is capable of screening 20,000 compounds. And we have some other chemical compound libraries. Importantly, a long time ago, we actually purchased 2 genome-wide RNAi libraries, siRNAs, which are synthetic small little oligos, and a lentiviral genome library for mouse. So we have the capability, if you want to do smaller, large-scale genome-wide screens or more targeted screens with the lentiviral RNAi approach, or just the siRNA approach. So we have the capability of doing those. Importantly, we also have the capability, if you just want some targets from those libraries, we can give those to you. You don't have to worry about ordering them separately. You can come to us and we can give you out those constructs to be able to do whatever you might want to do for your research. This is just an example of a paper that came out a couple of years ago that we supported. It's not in cancer, but it's just a good study to show what we're talking about. They screened 20,000 compounds using this set to identify specific targets and structures, and they designed found a selenomycin, which is a naturally occurring compound that had the structures of interest, and they showed some nice observations about how that could impact cardiac remodeling. And so just to highlight that, if you're interested to know more about what that is or how you might be able to use that, that's a good study. And just for context, we always ask every year, what are some services that the community might want? And I'm just listing here the other services from the most recent survey that people asked for, and how many people actually either had specific sub demands for those things. But really the point here is just to show you, here's what the community has asked for, and we're evaluating those. So if you in particular have a desire for one of these particular things, or if there's something else and there's other needs that we don't currently have, or they're not on the screen, they're important to think about, please let us know. Part of these lunch and learns are for us to give you information, but we also want information from the community outside of the normal survey. And the surveys go to faculty. And so for the graduate students and postdocs who are in the room, it's your chance to kind of give us some flavor of what you might be interested that may or may not align with the general faculty community. With that, I promised you it would be short. I didn't lie. I'm happy to take any questions. I'll stay here for a little while if there are no questions. People just want to chat. There's more food to have, but I'm happy to address any questions or talk about anything you're interested in. Yeah.
And those prices are high.
Yeah.
Price that it's not a moment supported. This is because it's kind of could maybe fit in the histology. I mean,
Yeah.
histology. It's probably historical reasons why. But is that any plan for that?
We have heard from several investigators of that need. Historically, there haven't been a lot of cancer users using it. And so that's why we haven't really evaluated it. But the cryo-EM was the opportunity to rethink whether or not we should incorporate some of those things. I can't say whether or not it will in the short term, but we have heard that same feedback. But yeah, It is, it is expensive. I mean, the technology is an expensive technology to maintain too. So that's also a part of it is for what it's worth, we have a limited budget to subsidize all of these things. And even that CCSG grant, if we get it, it's a small grant. It's not like a large facility. And so we have to be careful what we can spend it on. And none of the CCSG funds go to spend what we talked about today. So that's all additional amount of money that has to come up with to support that too. But we have heard that. Yeah. And part of it too is a lot of in the field, EM is moving where there's less EM locations and facilities, and they're moving to the more national labs because the national labs have much better, much stronger microscopes, and they're actually becoming cheaper. The wait time is an issue, but a lot of the EM facilities in particular across the country have been moving to that model of just being the outsource kind of conduit to those facilities. And so we have not fully evaluated what the cost of that is yet. But that alone is cheaper than bringing him in-house. Any other questions? Okay. Well, if anything comes up, again, I strongly encourage you to reach out to any of the individuals from those shared resources. They're all very nice people. They like to hear about your science and see where they can help. And then if you have any feedback you want to give, not publicly, and you want to email, feel free to do that to Tracy and I as well. Okay, thank you.
Feel free to take more food.
I'm so sorry, this meeting, I have no idea what happened, it's on my calendar, I'm back